So most flowering plants are hermaphroditic and accrue fitness both through pollen export and through pollen receipt. Several potential advantages of these dual sex functions have been proposed. For example, self-fertilization can provide reproductive assurance when pollinator services is unreliable or when population density is low. Hermaphroditism may also provide efficient allocation of reproductive resources in those situations where there is low marginal return from additional investment in a single sexual function. Today, I will explore another potential advantage of hermaphroditism. Hermaphroditism may increase mate diversity if mating portfolios differ from male and female perspectives. Such an increase in mate diversity may be adaptive if it increases the likelihood that offspring will be successful in spatially or temporally heterogeneous environments. This figure shows foraging itineraries for two bumblebees foraging through a population of single-flowered plant. And we're going to look with reference to a particular focal plant, and you can see that as these bumblebees forage through the population, flowers that are visited immediately preceding the focal plant are potential pollen donors of this flower, whereas after the bumblebee, each foraging bumblebee visits the focal flower, flowers visited subsequently are potential pollen recipients. So what I've just described is the potential for mating through these two functions, but what we lack are detailed accounting of mating portfolios in flowering plant populations. And part of the problem here is that nearly all work on mate diversity in flowering plants has focused from the female perspective. So there's been a lot of work on multiple paternity where the focus is how many mates sired seeds within fruits or how many mates sired seeds on maternal plants. But by contrast, we know almost nothing about mate diversity from the male perspective. In other words, how many individuals, pollen recipients in the population were sired by a particular hermaphrodite in the population. And so my talk today is going to explore, do mating portfolios differ from the male and female perspective? If they are very different, then potentially hermaphroditism is increasing mate diversity. Whereas if they broadly overlap, then there's not much increase in mate diversity associated with male function. So this talk is part of a series of talks you'll be hearing, the next 4 talks in this session. My group and Randy Mitchell and some of our collaborators have been, for several years, exploring how genetic and ecological factors influence mating patterns, focusing on this species, Mimulus rhingens. This is a wetland species from eastern and central North America. The flowers last for just a single morning. They anthracene hiss at 5 a.m. By noon, throughout almost all the populations we've sampled, all the stigmas are closed. They're pollinated primarily by bumblebees. Unlike other members of the genus, Mimulus rhingens, the stigmas are not touch sensitive. So after an initial bumblebee visit, they gradually start to close. That can take anywhere from 30 to 90 minutes usually. During that window of time, flowers will get between 1 and 3 visits. So they're getting a limited number of visits, and this is important when you're thinking about mating portfolios. It's not as if they're getting large numbers of visits. There is wide variation in male fertility and functional gender, and in the talk immediately following me, Dorothy Christopher will be talking about Bateman's principle. I should also mention that there's wide variation among populations in selfing rates, and we also see wide variation in floral display size from populations with just typically 1 flower to populations with many flowers. And Wendy Semsky and Randy Mitchell in the following two talks will be talking about genetic and ecological factors influencing selfing rate variation in this species. I should also mention that pollen carryover is very limited in mimulus ringens. So we've looked at this two different ways. One way that we've done this is to mark focal flowers with fluorescent dye particles and then look at how many flowers successively visited by a bumblebee receive fluorescent dye particles. And so if you look at the yellow curve, you can see that most fluorescent dye particles only disperse to the next 4 flowers in the visitation sequence. We've also done this with paternity analysis, and when we look at pollen-mediated gene dispersal, again, it's very short distances. So that's the blue curve here, and we see that most pollen-mediated gene dispersal is to the first 4 flowers in the visitation sequence. Now this turns out to be important because with this very limited pollen carryover, this actually increases the likelihood that mating portfolios will differ between the male and the female perspectives. Okay, so the approach that we undertook to quantify mating portfolios, just take a single snapshot in time. So this is a single day quantifying mating events for each individual in an experimental population. Now the source of the plants for this experimental study were from a predominantly outcrossing population that typically has single flower displays. Nearly all individuals have single flower displays on a given day, and these plants typically only make 6 to 8 flowers over the season. And again, this varies widely amongst populations, and Wendy and Randy will be talking about this. So we set up an experimental population, and we allowed natural bumblebee pollination at this site. In mimulus ringens, all flowers mature into fruits, and we have very high germination rates, typically in excess of 90%. So for every single progeny array, we then genotyped 5 seeds from each fruit, and with an exclusion probability, and it genotyped them in 8 microsatellite loci. With an exclusion probability of 0.98, we were successfully able to assign nearly every seed. 240 out of 245 seeds could be assigned to a single sire with greater than 80% confidence, and in fact, we assigned the majority of the seeds to a single sire with 95% confidence. Okay, so this is one way of visually presenting each, or in this case I've shown you so far 10 out of 49 hermaphrodites mating partners through both male function and through female function. So just to walk you through what is being presented as a bipartite network. So each of these colored bars is a hermaphrodite, and so they're represented for their male function and their female function. And the curves then show which of the 49 donors they mated with through both male function and female function. And this can be seen a little more clearly when we look at these hermaphrodites just one at a time. Okay, so here we have hermaphrodite number 30 shown in green, and it not only has received it not only self-sires, so it transfers its own self-pollen to fertilize its ovules, but also sires seeds on 3 pollen recipients in the population. And this same hermaphrodite, through its female function, has its seeds not only self-sired, but sired by 4 outcrossed donors in the population. The important thing to note here is that the identities of these pollen recipients that this hermaphrodite is siring differ from the identities of the sires who are siring ovules on this hermaphrodite. Okay, I'll show you one more example. So this is hermaphrodite 38 shown in gold. So hermaphrodite 38 sired seeds on 7 pollen recipients in the population. And 5 pollen donors sired seeds on this same hermaphrodite through its female function. Note, in this case, we find one case where there's reciprocal mating. So hermaphrodite 38 mates with hermaphrodite 310, both as a male and as a female. But again, you can see that for the second example, that there's very little overlap through the 2 sexual functions. If we look at this and integrate across all 49 individuals in the population, what we see is that mates usually differ from male and female perspectives. And across 49 plants, the mean Jacquard similarity between outcross male and female mate composition was just 0.06. So in conclusion, dual sex roles increase mate diversity in hermaphroditic flowering plants. And this will especially be important for species, or at least populations, that have very few flowers over the flowering season. It'll also be important for species that have very limited pollen carryover. And this finding may help explain the maintenance of hermaphroditism under conditions that would otherwise favor the evolution of separate sexes. And with that, I would be happy to answer any questions. Yeah.
Do you have any downstream data on fitness?
So it's a fascinating question. You know, what is really, we don't for the study. So what is really needed are studies that look at the success of individuals siring in elsewhere in the population, right? People have, and I've done some of these experiments where people have done SIB competition experiments. So you look at genetically diverse versus full SIB arrays, and then you look at them in different environments. And that work has been done, but no one has done this from the male perspective and looked at whether in heterogeneous environments, is your siring success in those different environments allowing you to, you know, is there bet hedging associated with the variable environments? And I don't know of any of that work. And Spencer and I were talking about this earlier, and I think that's something which is really needed. I'd love to see it. Yeah.
So you said the stigma closure isn't touch sensitive. Is it known what makes it close? Do you think it's light or temperature?
So I have 2 hypotheses. So one hypothesis is that well, it is triggered by pollen being present, and so pollen is germinating. The question is whether it's just the presence of the pollen germinating, or whether it's also affecting sort of the hydration of the stigma as all this pollen lands on it. And we've never teased that. Spencer?
Have you got any thoughts on how mating portfolios might be affected by clonal propagation? Your species does have the capacity in natural populations for cloning.
So, yeah, so Mimulus ringens does, because we've actually looked at genetic structure of populations to the extent of cloning, we get clumps of plants. We don't get large clones. So the more